morning, dear students. Today we are going to start our class with the topic Mode of Infection and Pathogenicity of Plasmodium. Regarding its taxonomical details, it comes under the phylum Apicoplexa, class Aconoidacida, order Hemospororida, family Plasmodidae, genus Plasmodium. So, what is this Plasmodium? Plasmodium is a parasitic protozoan and the causative organism of malaria. Then, which is the vector of this Plasmodium? Vector is female Anopheles mosquito. Female Anopheles mosquito is the vector of Plasmodium protozoan parasite. Then there are several different species exist in human. Which are the species that cause malaria in human being? They are Plasmodium vivax. This Plasmodium vivax is responsible for causing the most widespread form of malaria in human being. Then Plasmodium ovale is there. It's uncommon. It's not that much common in human being. Then next one, Plasmodium falciparum. For this one cause the most severe symptoms associated with the malaria, because that means when this Plasmodium falciparum affect human being, the symptoms occurs because of that Plasmodium is very severe. And then two more species also there, Plasmodium malariae and Plasmodium knowlesi. So these are the species of Plasmodium that is causing malaria in human beings. In chimpanzees also this Plasmodium protozoan parasite exists. They are Plasmodium ricinovi, Plasmodium gaboni, Plasmodium falciparum. In the case of reptiles, there exist two varieties of Plasmodium. They are Plasmodium mexicanum and Plasmodium floridans. In the birds, another two varieties, Plasmodium relictum and Plasmodium juxtanucleae. So it's just for our knowledge. Several different species of Plasmodium exist in different vertebrate hosts. So let's have a deep understanding about the life cycle of Plasmodium. Plasmodium alternates between vertebrate and mosquito host during its life cycle. And in its life cycle, its a sexual phase happens inside the mosquito. Then which is the transmissive stage of this Plasmodium? It is called sporozoid. And these sporozoids are present in the salivary gland of infected mosquito. When this mosquito bite a human being or a host, along with the anticoagulant saliva, this sporozoid enters the host body, and on reaching the host body through the bloodstream, it will reach liver. If the host be sometimes instead of human, if it is a bird or lizard, then that time this sporozoid is reaching endothelial cells or macrophages. Leave that, but here it's the case is human. So here the sporozoid enters the liver, and on reaching the liver, it somehow managed to enter inside the liver cells. And inside the liver cell, it goes on several rounds of multiplication and will change to another form. It is called the merozoites. And this merozoites later is released out of the liver cell to bloodstream again. And that merozoites then will attack an RBC cell. So there is two more terms we need to be familiar. Term means a phase, that is pre-erythrocyte phase. Pre-erythrocyte phase means the phase before it infect an RBC. That means the time period before the infection of an RBC. That phase is called the pre-erythrocyte phase. or it is otherwise called exo erythrocyte phase. After this phase, these merozoites attacking RBC cell, and it managed to enter inside the RBC cell, and after getting inside the RBC cell, it undergoes several rounds of multiplication and proliferate inside the RBC, and then finally rupture that RBC, and all the merozoites are released outside, and it is free to attack another RBC. So that cycle repeat, and this phase is called asexual blood cycle. And while the time an RBC cell rupture and the merozoites are released, then that time some toxic substances also released to the outside, and that may lead to develop high fever in the host. Okay. At that time, some of the merozoites they are changed to gametocytes. Gametocytes means female and male gametocytes. These female gametocytes are called macrogametocyte, and male gametocyte is called gametocyte. In this form, when another mosquito come and bite this host, then these two gametocytes can enter the body of this mosquito. And on entering this body of this mosquito, this microgametocyte, it multiply asexually and will form into a flagellated microgamete. This microgametocyte, on reaching the body of another mosquito, it undergoes several rounds of multiplication and assume a flagellated form. It's called flagellated microgamete. And each flagellated microgamete can fuse with the macrogametocyte and can form a zygote. And this zygote later developed into a motile eukinete, and this motile eukinete pierces the gut wall of the mosquito, and after piercing the gut wall, it forms a cyst and accumulates, and it settles inside that cyst, and that stage is called oocyst. And inside the oocyst, it continues, goes on so many rounds of asexual multiplication and increase the number, and later form the motile sporozoites. Then this motile sporozoites later is released out of this cyst to the bloodstream of the mosquito, and from the bloodstream it again reaches the salivary gland of that mosquito, and get ready to inoculate to the body of another human being to continue the cycle. Okay. Next, we are going to see more details of that asexual blood stage. What is asexual blood stage? That I already explained to you in the former slide. That is, that stage means the during the time that merozoite attacks RBC and undergoes so many rounds of multiplication and proliferation. That phase, and again rupturing that RBC, and again it is going to attack another RBC. That is that cycle, asexual blood stage. So the some details of that stage that we are going to understand now. For that, first thing we need to learn more thing. What are the features of a merozoites? Its picture is shown here. Inside the RBC, it undergoes so many changes. That changes are shown here. Merozoids first it changed to a ring form, then a trophozoid form, and later a schizone stage. All things we will learn in the following slides. Anyway, first we can learn about the merozoids. It is the inversive form and it has some specialized organelles. It has some specialized organelles inside it to invade the erythrocyte without lysing them. Without lysing the erythrocyte, it need to invade the erythrocyte. So, what all structures inside the merozoid helping it to invert the erythrocyte that we are going to see. Looking to its shape, we can see that its anterior end is like a prominence. Anterior end is like a prominence, and it is supported by three cytoskeletal rings. They are called polar rings. They are called polar rings. And anchored to this ring present two sets of secretory organelles. The first set, it is called rhopteries. Where it is? Rhopteries. First set of secretory structures is called rhopteries. And similar to that, a small means shape-wise, they both type of granules, secretory granules are similar. Even then, its size-wise it is small when compared to the size of this rhoptery. Both are pear-shaped. Both are pear-shaped vesicles. And the first big one are called rhopteries, and the small pear-shaped secretory granule is called micronemes. Micronemes. So, two sets of secretory granules are there. It is attached to this microzoid in such a way that it can easily secrete its contents to outside as this microzoids invade the erythrocyte. Okay, and we can see some rounded vesicles here. They are called dense granules. They also contain some chemicals inside that also will secreted after invading the RBC or erythrocyte. And similar to this dense granules, there present some elongated such type of granules. They are called exonemes. What its purpose? It is used by the merozoid to exit from the schizones that we will see later. Then coming to the surface of the merozoid, its outer surface is covered by a thick bristly coat, and underlying to that coat present three membranes, and those three membranes are collectively called pellicle. The outer, among that three membranes, the outer one is called plasma membrane, and the inner two together form the inner membrane complex. Inner membrane complex. Then what is the significance of this inner membrane complex? This inner membrane complex contains the actin-myosin motor. The actin-myosin motor that propels the merozoid while it invade the RBC. Also, there present some longitudinal microtubules that we can see here. Longitudinal microtubules. It is attached to the inner layer and anchored anteriorly to the polar rings. And also we can see a nucleus, single nucleus, a mitochondria also there. Then epicoplast. Epicoplast means it is a vestigial plastid that usually present in some parasite forms. Some ribosomes also we can see. These are the minimal equipment that is present inside the small tiny merozoid that is helping it to invade the RBC. Okay. So now we are clear about the detailed structure that present inside the merozoids. So now we are going to see how this merozoid invert the erythrocyte. It is a multi-stage process. It's a multi-stage process. Let's see what are the process that is happening for the invasion of the erythrocyte. For that, first an interaction is required. Interaction between some adhesive protein that present, that secreted from the micronemes. Micronemes means it is a secretory granule that we have seen in the former slide along with the rhabdoids. So these micronemes secrete some adhesive protein. It makes an interaction with the glycophorins that are present on the erythrocyte membrane. So through that interaction, these merozoites come closer to the erythrocyte surface. And while the time of that association, this parasite deploy multiple receptors. It deploy multiple receptors. Why deploy multiple receptors? The purpose is that depending upon genetically variant hosts, it need to maximize the chance of invasion. That's why it deploy multiple receptors. And these receptors later are tested as immunogens. They are tested as immunogens. And against that, later vaccines have developed to treat this disease. And after the first association with the erythrocyte surface, then these merozoites reorient. It will reorient. Why it reorient? To bring the apex portion of the merozoite in contact with the erythrocyte membrane. What is the purpose? Then only the secretory granules can effectively secrete its contents to facilitate the inversion. And through that reorientation, it makes an irreversible close junction between the erythrocyte. And in the meanwhile, the rhabdoids secrete its contents, and with the help of that, on the surface of the erythrocyte, a deep membrane-lined pit will form, and to that pit, that parasite moves into the erythrocyte. After the parasite moves into that pit, that erythrocyte membrane closes. It closes over that pit, and by that way, now the merozoite is trapped inside that membrane-lined space. By that way, it is entering, invading the erythrocyte. Let's see a picture of that inversion. This is the picture. Here first it's coming and it make an association, reorienting, and later a pit is forming and it is moving into that pit and it is completely enclosed inside and come inside the erythrocyte, and later it is coming to this stage that we will see in the coming section. It's a ring form. In this stage, that merozoite appears in the form of a ring because of a thin portion in the center. Okay. That's about the invasion of the erythrocyte by merozoite. Next, after invading the erythrocyte, what are the changes is happening to the merozoite that we are going to see next? After entering the erythrocyte, merozoite loses all its invasive organelles. It loses all its invasive organelles. It lost. And it become a discord couplet. I have already shown you in the former picture. It become a discord couplet with a thinner central region. And as we observe it in this stage under the microscope, it appears in the form of a ring. So that stage is called a ring stage. And from that stage onwards, it start to ingest the erythrocyte cytoplasm and start to degrade the hemoglobin of the erythrocyte. Okay. Then this parasite, that disc-shaped, cup-shaped parasite, then transform to a plumber trophocyte. A plumber trophocyte. You can see here. Plumber means it's more, it become more spherical, bulged one. So that's a plumber trophocyte, and start to feed more voraciously, and it start to break down the hemoglobin's heme and convert that to a malarial pigment. Start to convert that one to a malarial pigment. Its name is hemozoin that we can see here. This is hemozoin. See here. And Then it enters the next stage, that is the schizone stage. At this stage, this parasite's nucleus start to divide repeatedly, and the number of division, how many division it is undergoing, that depends upon the species. And after that division of nucleus, each nucleus enters a merozoid bud form, and it assemble all the required organelles also inside it. And when the merozoid bud mature, then it detach from this parent schizone stage and leave them by lefting small amount of cytoplasm. Its name is residual body, and it contains the hemozoin. Finally, from the exonym means it is also dense granules. Dense granules, it is filled with secretions, so many enzymes, protease substance. So, as release its contents, it contains so many proteases. And with the help of these proteases, a series of chemical changes happens, and as a result it break the merozoid, break the erythrocyte and release to outside, and now it is ready to infect another erythrocyte. Okay, that's about the changes that happen to that merozoid after invading the erythrocyte. Next, we are going to see the sexual stages that happening inside the mosquito. First, we need to learn about the gametocytes. While being inside the body of the host, some of the parasite at merozoid stage, they develop to either male or female lines, and they are having their own characteristic structure, but they have identical chromosomal content. Chromosomal content be identical, but their structure wise there will be some difference. And most oftenly they appears like curved forms, but in some species, the gametocytes of some plasmodium species, they look spherical. And two genders, male and female, they differ from each other with respect to their cellular detail that I already mentioned to you. And even then they have some common features, like they possess some long microtubules along their body that helps to define their shape, and that will varies from species. And also this mature gametocyte possesses a disc-like disc-like osmophilic body in both male and female gametocyte. What is its significance? It contains chemicals. They are secretory vesicles. What is the function of the content of this secretory vesicle? It is helping the this parasite to exit from the host cell, and that gametocyte entirely is surrounded by three membrane pellicle. It is surrounded by a three membrane pellicle. That's about the different features of gametocyte. And when a mosquito come and suck blood from that host that are having this gametocyte, male and female gametocyte, then this gametocyte enters the body of the mosquito. On reaching the body of the mosquito, this gametocyte escape from its surrounding erythrocyte membranes, and it get triggered. Why it triggered? Because of the temperature drop, and the presence of certain chemicals in the insect's body. This gametocyte get triggered, and as a result, that male gametocyte, it divide rapidly and form gametes. And this gamete, it will sprout long motile flagella. That process is called exflagellation. After exflagellation, each male gamete is capable to fertilize the female gametocyte to form the zygote. Okay. Next, we are going to see the asexual stages that is happening inside the mosquito. After forming the zygote inside the mosquito, then this zygote undergoes meiotic nuclear division and it elongates. Before in the zygotic stage, it is somewhat in spherical form. After meiotic nuclear division, it elongates to form a motile eukainate. This is somewhat its structure. And in the form of the motile eukainate, it develops numerous micronemes. We know that micronemes are the secretory granules. And also polar rings also they are developing, but there is no rhopteries. Rhopteries are absent. And inner membrane complex bearing the meiosis, that also lacking in the motile eukaryote. So that's about that stage. Then after that stage, using the enzymes present in the micronemes, this eukaryote penetrate the gut epithelium of the mosquito. You have to understand that all these stages are happening inside the mosquito as it suck blood from blood from an infected human. Okay, so because of the help of the enzymes that present in the motile eukaryote, it penetrate the gut epithelium of the mosquito. And on reaching the gut epithelium, that parasite get rounded up. It secrete a cyst and occupy inside that cyst. And that stage is called oocyst. I will show another picture of that. This is the oocyst stage. This eukaryote is penetrating gut wall and it create a cyst and encysted in the gut epithelium. So that is the oocyst stage. Then next, which are the other changes that is happening after this oocyst? Within the oocyst stage, this parasite replicates its DNA. It replicates its DNA many times, and as a result, a massive nucleus will form here. And as a result of that division, so many rounds of DNA replication, so many genomic centers, means so many copies of its entire genetic content will form inside that oocyte. So that we can see, this is the oocyte and DNA replicates many times and form a massive nucleus. Massive nucleus, that means it contains so many centers, means so many copies of its genome because of this internal mitotic division. Then after that, these groups of chromosomes, it separate and form separate nuclei around the parasite periphery. It moves to the periphery of the parasite and later it will move into some finger-like projections at the parasite surface. That I will show you. That nucleus is moving to the periphery and it get located. It move to some finger-like projections that's forming on the parasite surface. And these finger-like projections later it develop new organelles and detach as separate sporozoid. Separate sporozoid. And when mature, they migrate through the cyst wall and will reach the insect's blood cavity that I will show you in a picture. So when mature, that sporozoid it released to the its blood of the insect and later it will migrate and can come to the salivary gland of this mosquito to inoculate another host. So let's see what are the features of this sporozoid. In this sporozoid, we have already seen the structure of merozoid. So compared with the merozoid, sporozoid is somewhat elongated one, but structural plan is somewhat similar. They are elongate and slightly curved cells, especially in the case of Plasmodium falciparum, and its length is 10 millimeter long, tapering at both ends that we can see here with a central nucleus. And apical organelles, so many apical organelles also present. Polar rings also we can see with a set of rhopteries. And then to the inner membrane complex. Inner membrane complex means those membranes sit below the plasma membrane, seen below the plasma membrane. And it is attached with a set of longitudinal subpellicular microtubules. Longitudinal subpellicular microtubules. And also this tubules is stretching back from the polar rings that we have seen in Merozoite itself. So from that polar rings, these microtubules are stretching back and create a spiral cage-like cytoskeleton around the parasite. Okay, and also along with all these structures, we can see a mitochondria, and apicoplast. Apicoplast is a vestigial plastid. They both we can locate somewhere towards the posterior end. And like the merozoid, its apical organelles means maybe that secretory granules. They contain bacteria, means so many enormous number of invasive proteins and adhesins. Example, circumsporozoid protein. Circumsporozoid protein and thrombo spondin related antigen protein. These are some examples of some adhesion protein. And what are its purpose? It enable it to attach with the host glycosaminoglycan receptor for that association. And these adhesion proteins are now considered as important candidate of immunogens to develop vaccines against this malaria. Okay, clear? That's about the details of the life cycle of the Plasmodium. I hope that's very clear to you in full depth. Next is about the pathogenicity. It causes the disease malaria that we already have learned from the beginning of this topic itself. What are the symptoms? Symptoms include high fever, headache, and chills, shivering, and that will appear 10 to 15 days after the bite of infected mosquito. And during that time, in the initial phases, it will be mild and so difficult to recognize as malaria. So that is the main crisis in this disease. If we left it untreated, if that bite is happen from a mosquito that is infected with the Plasmodium falciparum, then it will progress to severe illness and death will happen within a period of 24 hours. So death also may occur because of this disease. Then how we can prevent? Easy thing, through controlling the vector. Vector means the female Anopheles mosquito. By controlling its population, we can control this disease. And by usage of medicines, that is preventive chemotherapy. It uses medicines. Some medicine means individually, and also sometimes we apply combinations of medicines to prevent the malaria infections. Then vaccine also there. RTS S bar AS01 malaria vaccine is a common one. It is administered among children in those regions where the infestations of Plasmodium falciparum malaria is more. Its transmission rate is high in those regions. This vaccine is given frequently, and this vaccine is that much effective to reduce the malaria, and especially in the among the young children. So by this way, I think regarding the life cycle and pathogenicity of Plasmodium is clear to you. So we can wind up today's section. Thank you.
I'm an icon, man. Damn, my girl about a 10. I should fuck her in the Benz.